Hello, welcome to today's lesson, monoclonal antibodies. For this lesson, you will also need the attached Word document, which is about monoclonal antibodies, and also the starter, antibacterial agents. I would like you now to start with today's lesson and do task 1 of the starter, antibacterial agents. I assume you've now completed the starter. Before we proceed with today's lesson, if you are unsure of any of the following terms, you should take a moment to revise them. The terms that you need to revisit are first line of defence. Do you remember what the first line of defence agents are in the human body? Don't forget plants. We also have to know about plant biology. So take a moment also to revise first line of defence mechanisms in plants. Key terms that you need to know for today's lesson are antigen, T cells, B cells, antibodies, phagocytes and phagocytosis. If you aren't aware or cannot remember these terms, you should take a moment to pause this clip and revise these terms. Lesson is monoclonal antibodies. It's describing the production of monoclonal antibodies using lymphocytes, making a hybridoma cell and their ability to produce antibodies, explaining the uses of monoclonal antibodies, which include pregnancy testing, diagnosing for blood clots, cancer cells and cancer treatment and HIV, and the advantages of monoclonal antibodies to specific target cells rather than using drug and radiotherapy treatments. So you should have by now completed the starter. Here are the answers for task 1. The answers are also in the worksheet, and there are other tasks in the worksheet from the infection control module. Recap. You should now pause this clip and match the definitions. These are from the infection control module you've done in year 1. Answers for the previous slide. If you weren't aware of them, don't forget to take time to actually revise them. So I'd like you, using the accompanying PowerPoint, to watch the clip Immunity. Some of you may recall this from previous lessons. You need to understand what are lymphocytes, how do they invade pathogens and how do they recognise them, what can lymphocytes do. If an antibody, a B cell, does not destroy the pathogen directly, what else may help destroy the pathogen? And here are the answers which accompany the clip. Lymphocytes are white blood cells which are produced by the body. They are antibodies. Antibodies are produced by the body after first line of defence mechanisms have failed, after first line T cells defences have failed, and cells such as macrophages and phagocytosis, where the macrophage tries to eat and engulf the invading pathogen, have failed. T cells have a number of cells which they summons the antibodies, the B cells, to latch on to recognised antigens on invading pathogens. Antigens are special surface proteins which pathogens carry, and the body can make millions of antibodies which will latch on to a recognised surface antigen. Remember, these are proteins, and proteins have a lock and key mechanism, so only a specific antibody can lock on to a specific antigen. Lymphocytes obviously will lock on to this invading pathogen, and the aim is to destroy it. It will clump together the pathogens and it will undergo a process of phagocytosis where it will engulf the the pathogen, and because the white blood cells are multi-lobed in their nucleus, it can totally engulf it and break down and destroy the pathogen, where the rest of the body, the immune system, will go on to finish off the destruction of the invading pathogen. This is why we feel so ill on the first few days of any cold, flu or infection, because our antibodies are working, and it is them working which makes us have a fever, makes us feel lethargic, that's tired, makes us ache, because your system is developing these antibodies to destroy any invading pathogen. You should take a minute, if you haven't already done so, to revise the terms for first line of defence in the body. I will pause this clip now and allow you to do some revision time. Obviously you can continue with the PowerPoint, and you can obviously revise these points at another time. Some of the points for first line of defence are the skin, lysosomes, and chemicals in certain parts of the body, moist areas of the body. You've got stomach acid. You've got obviously scabs which form on cuts. So these are just some of the terms. There are several of them when we cover this particular topic. So I've not detailed all of these, so you should make sure that you're aware of them. So obviously when the body is first attacked, hopefully our first line of defence will help prevent that infection invading our bodies. Do not forget, we are subject to millions of different types of pathogens every day, and our bodies have already coped with a mechanism to help prevent you from becoming ill. Just think, do you not wash your hands, go to the toilet? You probably do, hopefully you do, but somebody else doesn't. So their gut bacteria will be on the door handle for the toilet. Their gut bacteria will be on taps. Their gut bacteria will be on light switches in bathrooms. Every time somebody coughs or sneezes, they have them nice wet sneezes that they sneeze into their hands. Do they wash them during the school day? No, but then they go and touch the door handle, the light switch. How about borrowing that pen of yours? So next time you lend somebody, think about, do they have bugs that I really want to share? So first line of defence has failed. The body needs to mount a new line of defence. It's imperative, obviously, we keep safe within our own immune system. So the antibodies occur from B cells. Now B cells are a result of being summoned by a T cell. T cells and B cells are all white blood cells which go on to make antibodies. So T cells, there are several. There's T1, T2, T helper. These are all special white blood cells which summon B cells, which are the antibodies. The T cells undergo a number of processes. The first one obviously is to do phagocytosis. This is when the white blood cells engulf the invading pathogen. It latches onto that antigen and it will break it down. That breaking down of that antigen, of that pathogen, is then obviously with a special T cell, T helper cells will summon the B cells, the antibodies, and the antibodies can actually latch on and destroy the invading pathogen, which has obviously been broken down. Now the body is actually capable of making over 2 million different types of antibodies, and these are all specific. That's why you might hear, oh, such and such has been off with a cold today. You might have already had that cold, and as you've already had that cold, you would have built up a resistance. That's because the antibodies for that code have obviously worked. After you've had that code, the body is able to actually create memory cells. These are special immune cells in the immune system. And if you have already had that code that your friend has now got, if they're obviously sitting next to you in the classroom or in a room with you, because your body has already been infected, it's created the antibodies. After the infection has gone, it leaves memory cells. You do not get that same code again. The problem is codes, flus, all develop a way of mutating. And that mutating means a new virus comes along, and then obviously you do not have the memory cells for this new virus. And this is why the body will start a whole new launch procedure again. T cells, white blood cells will summon the antibodies to actually destroy a new virus and go on to help you develop memory cells. In medicines, we have developed so many treatments now. We have obviously Alexander Fleming, who is able to make antibiotics. Don't forget, antibiotics are for treatment of bacterial cells only, not viruses. Viruses obviously are dead cells, or dead, they're not living. We say they're not living because they need a host. They need a cell to reproduce. If you can't remember the difference between bacteria and viruses, you need to take time to actually revisit what a virus is. So I said a virus is non-living. Viruses are very clever. We have a number of viruses. Viruses always have either a DNA virus or a DNA and an RNA virus, or just an RNA virus. RNA means that that cell, that virus cell, has already created its own mechanisms to start reproduction of new virus cells in the body. So flus and measles are DNA RNA viruses. They have these antigens on the outside, these special proteins. So when they latch onto your body cells, they need your cell to actually ensure that they can carry on that reproduction of their DNA, their RNA, their DNA. And we will be covering RNA and DNA this term. And this enables them to actually reproduce in the cells. If they do not have a new host cell, the virus cannot survive. We in science now, as a result of Fleming and his findings with regards to growing of bacteria all them years ago in the First World War going up to the Second World War, we have actually been able to now manufacture, and because science can actually look using electron microscopes at virus particles, we can take weakened virus particles or dead virus particles and create them into a vaccine. And then these vaccine programs in many countries are legal requirements that children and adults receive to help prevent issues. Can you think what benefits a vaccination program has? Obviously, you've all undergone vaccines at school for things like diphtheria, for measles, mumps, rubella. The girls would have undergone HPV vaccines. What are the benefits of a vaccination program? I'll give you a minute to think about that. Okay, your minute's up. So, I'll give you a few more seconds just to make some notes about what the benefits of a vaccine program is. Well, vaccine programs are taking, as I said, a dead or weakened pathogen, a virus particle, and injecting them into the community, into individuals. if there are sufficient numbers vaccinated, there are possibilities that it actually eradicates in the community killer pathogens. Obviously, to date, we've been able to eradicate smallpox. We are working on, and we have nearly eradicated polio. Bill Gates, obviously the founder of Microsoft, has started a worldwide polio vaccination program in endeavouring to actually eradicate polio. We are also hoping to try and eradicate cases such as diphtheria and whooping cough. The numbers of these cases in the UK and in the world are dropping, and hopefully one day we will be able to eradicate these. These have long-term cost benefits for lives. People do not become ill. We often hear of these conditions such as whooping cough and measles. Measles, we assume, is a relatively mild condition, a childhood illness, but unfortunately it is not. It can cause death. If people do contract things like measles, they can end up with deafness and other debilitating conditions. So they are serious illnesses which need to be prevented. So long-term health benefits are significantly increased, and also that has a cost effect for the health service. So you should now take time to pause this clip and read the attached Word document on monoclonal antibodies. In 1984, so this is a recent scientific development, Dr. Milstein and Dr. Kohler discovered a way of making a pure antibody in a test tube. So please pause this clip now and read the attached document, The Making of Monoclonal Antibodies. Okay, before I go on and you answer the question, the discovery of a magic bullet, monoclonal antibodies are a form of medical treatment which rely on the body's immune system. Monoclonal antibodies are proteins that only target specific cells or chemicals in the body. Now, it uses the mammal's lymphocyte system, which stimulate the body to make antibodies. When obviously these cells are collected or harvested, for another term, they can be purified. When making monoclonal antibodies, we use mice antibodies when we make them and purify them. And we can collect these antibodies which are specific to only one antigen or chemical. And when we make these monoclonal antibodies, they will then only target a specific antigen within the body of the person they are treating. These can include tumours, specific diseases, or even chemicals and hormones which the body make. That's why we can use them in pregnancy tests. That's why we can use them to detect cancers, the level of cancers, and also infections such as HIV. So these monoclonal antibodies can be made with a marker, and these markers actually highlight when a blood test is taken how seriously ill an individual is. So for example, we use them to detect prostate cancer. So we inject these monoclonal antibodies with a marker into individuals. We leave it for a set length of time, and then we collect another blood sample, and the blood sample will be marked with the monoclonal antibody, and we can determine at what stage an individual is within somebody's illness for the specific targeted antigen that they have been injected with. So we're using them more and more these days for conditions such as cancer, HIV and syphilis, which is obviously HIV and syphilis are sexually transmitted diseases. Now, when monoclonal antibodies are used to treat a condition, they will only target specific cells. This then triggers the immune system to recognise the target cell and destroy it. This is why it is useful in cancer treatment, because it will only target the infected cell. Unlike other treatments for cancer. It will not travel all around the body through the bloodstream, destroying other cells. It will only target the specific antigen it is required to. When it targets the cell, the receptor on the cell that it is aiming to destroy, it prevents that cell from dividing. And what we can also do is when we inject the monoclonal antigen, the antibody, it will be laced with, or it will be manufactured to carry a toxic drug which will destroy, or a radioactive substance which will destroy the actual infected cell directly. And this actually prevents damage to unaffected body cells. So now you've got that particular information, why, using the information leaflet, was the discovery of monoclonal antibodies called or labelled the magic bullet? Okay, you should have come up with the fact that they had found a way of making antibodies in a test tube, in vitro, in glass. And obviously this is a tailored weapon that can home in and destroy a cause of a specific disease. This obviously was made in a laboratory. They are artificially produced. It improves speeding up research and diagnosing, treating many different diseases. And obviously when we look at things mono, we look at the suffix mono. Some of the key words and terms in science in English language allows us actually to understand what the term means. So heme, H-A-E-M, means blood. So the suffix heme is relating to the blood. In the case of mono, the suffix means only one. So a monoclonal antibody is only one colonal, one colony of targeted treatment to attach to an antigen. So it's one colony of antibodies which will attach itself to one antigen, or a bacterium or a virus, and then it will actually destroy and seek out and destroy that infected pathogen. This increases the speed of any treatment and reduces damage to an individual. You should now take time to watch the attached YouTube clip on explaining monoclonal antibodies. So monoclonal antibodies are manufactured in a lab. They take a specific antigen. In this case we use mice. So we take obviously the B lymphocytes, which actually make antibodies. Now obviously B lymphocytes do not divide, so the division would have actually happened before we take the B cells. The B cells make millions of different antibodies. So they'll take a specific antigen and from the rat or a mouse, and we can take from rats and mice because they have a very similar DNA pattern to humans, and we can actually take antibodies from them, and we can then produce them and grow them in a hybridoma cell. Now, when we take their antibodies, we take this hybridoma cell, which we're manufacturing in a laboratory in the test tube, and we can take a tumour cell. So we know certain tumour cells have certain protein makeup, and science has discovered that we can use the proteins from these tumours to determine that an individual is carrying certain cancers, certain infections. So we take an antibody from a mouse, one that has been manufactured, we've injected that mouse with a certain condition, and we can harvest their antibodies. We can take the tumour cells, and what we can do is we can put both of these cells into a hybridoma cell. This cell goes on to divide. So we've fused the antibody from the mouse, and we've fused the tumour cells in this hybridoma cell, and it makes this what we call a hybrid. So it then allows in this hybrid cell clones of specific cells with the actual antibody from the mouse to divide. This cell then we can harvest the specifically made antibody, which takes the white blood cells, the B lymphocytes from the mouse, and the information from the tumour, and creates a special antibody, rather like the memory cells that your body creates after you have had a vaccination. So we can now make a specific antibody using the infected mouse, which would have grown lymphocytes for the infected agent we want to grow in the test tube. And we then obviously take the tumour cells themselves from an individual, which obviously we can then still continue to grow in a laboratory. We pop them into a hybrid cell, and the cell goes on to multiply and divide and makes these special clones of cells, which then go on to make antibodies. And we can then take these antibodies and inject them into an individual. We can also use them for detecting in other ways. Oh, can you spot the mistake on this slide? I've only just noticed it. So, monoclonal antibodies is a complicated aspect of science. So you should re-watch the clip I've put in the video. You should read around it, read your revision book, and read the worksheet that I made, because obviously there's a number of treatments that we can actually use. We don't just use it for injecting, we use them for detecting proteins in urine, for obviously pregnancy testing, and we obviously now are using them for blood test markers, where we take infected blood, and obviously using monoclonal antibodies external to the body, we can use them for detecting conditions such as HIV. So you should obviously make notes in your revision what the benefits of monoclonal antibodies are, why are they effective, and identify how they can be used. I've detailed some of these on the information sheet. You need to know what the advantages and disadvantages of monoclonal antibodies are. And remember, if you have exam questions such as these, you'll only get full marks if you specify the advantage and a disadvantage. And then you should be able to take the diagram from earlier in the presentation to draw how monoclonal antibodies are produced. Obviously, some advantages for monoclonal antibodies are they only bind to specific cells. Healthy cells are not affected. In contrast, conventional treatments, when they are carried out for certain conditions, will be carried around the whole of the body and can target non-affected cells. So this can be detrimental to the individual. Radiotherapy targets affected areas of the affected cell in front and beyond tumours, so radiotherapy can actually do more damage to the body in trying to treat an individual. Another advantage of using monoclonal is that they can actually treat a wide range of conditions, but unfortunately the disadvantage of this is they are expensive to develop. They're not yet widely used. Initially they thought it was the magic treatment for a lot of conditions, but unfortunately using monoclonal antibodies or antibodies from the mouse caused unexpected side effects in humans. The mouse antibodies obviously triggered the human response system. Now, as obviously science has developed, doctors obviously are now aware of these side effects. They've been able to reduce some of the side effects from the manufactured monoclonal antibodies, but doctors are also now aware of these side effects. So as well as administering the monoclonal antibody, we can treat for the symptoms of the side effects. So today's lesson was monoclonal antibodies using lymphocytes, making that hybridoma cell, and for them to go on and produce specific target antibodies, and explain obviously the use of monoclonal antibodies. Obviously the pregnancy testing was detailed on the attached worksheet. Any queries, don't hesitate to contact me.